Hello, Bio9. So, real quick video here. I have some people asking questions, and you guys are all asking exactly the right questions. Sorry, I've been gone. I've been not stuck in the mountains. I've been happily camping. As you can see, I have post-camping hair. I haven't showered, and I don't even want to talk about how long. And, yeah, you're lucky that you're not around me right now. Anyways, so one of the things I wanted to show you guys, because this is a really important element of DNA science, is what nucleotide structure is like. Anybody who's taken one of my face-to-face courses knows that chemistry isn't my favorite subject, but I have great respect for chemistry. And unless we understand how the molecules that we keep talking about are put together, it's difficult to understand how they work with one another. So let's draw out, for funsies here, the basic unit of DNA and RNA, which is called a nucleotide. So we're going to start with the central portion of the nucleotide, which is called, in DNA, the deoxyribonucleic acid. So you have an oxygen at the top of the molecule, a carbon here, a carbon, 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 and then this carbon bonds back to the oxygen. We're going to number these carbons. One of the beautiful things about organic chemistry and biochemistry is that the carbons are always numbered in order in which they appear clockwise. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and as it turns out, there's a carbon number 5 sticking off of carbon number 4. In chemistry, or in biochemistry, we call these the 1', 2', 3', 4', and 5' carbons. If any of you have talked about the 5' to 3' orientation of a strand of DNA, this should start giving you an idea of why we call something 5' to 3'. So this is the central sugar. This is, right now, ribose. We're going to add some other elements to this. On the first carbon, on the 1' carbon, we're going to have our nitrogenous base. Okay, and for now, I'm just going to write base. This base, as you know, in DNA, these bases are A, T, G, or C. In RNA, though, on the other hand, let me get a different color here. Green and red, right? In RNA, you can have A, U, G, or C. T and U, U substitutes for T in RNA language, okay? So on the first base, on the carbon number 1, you're going to find the nucleic acid base here, all right? We're not going to worry about the shapes of those yet. On carbon number 2, you're going to find either a hydrogen. Actually, I want to do that a little bit differently here. Let me make this like our DNA or RNA. On the second base, you are either going to find a, what is going on here? I need my pen. Okay, I'm sorry, my pen is misbehaving. Oh, blast it all. All right, I don't know why my pen is doing this now. Hold on just a second. Okay, I think I have this working now. So, on the 2' carbon, you're either going to have the presence of a hydrogen for DNA or a hydroxyl, an OH. A lot of people call it OH, but it's called a hydroxyl if you're dealing with RNA. So right now, DNA is going to be coded in red, and RNA is going to be coded in green. That hydrogen is a very, very stable bond to the carbon there. Basically, what it does is it just stabilizes the carbon, and it just kind of hangs out. In the case of having an RNA nucleotide, you're going to have the presence of a hydroxyl here, not the hydrogen. Okay, so the hydroxyl, the oxygen will be bound to the carbon. This is a very reactive functional group. And so wherever you see OHs or hydroxyls, you know that covalent bonds can be formed there. Now, in terms of the third carbon, you're always going to find a hydroxyl here in DNA or RNA. Now, hanging off of carbon number 5, you can have 3 phosphates. Here's phosphate number 1, phosphate number 2, and I don't know why this is looking so ugly, and phosphate number 3. Now, does that remind you of any other molecule that you've heard of? Perhaps ATP? Ooh, ugly, ugly. Ugly TP. Let me erase that, hopefully. Okay, well, this is not cooperating with me today. I don't know why. My apologies. Come on, erase already. Okay. ATP. It's like ATP, but it's not exactly ATP. It does carry energy, and when you glue one nucleotide onto another, you'll lose 2 of these phosphates and maintain this phosphate to actually knock off this hydrogen. We'll get to that later when we talk about replication, and for now I just want you guys to focus on these elements of a nucleotide because, I don't know, it might be important in an exam question. So, for both deoxyribonucleic acid, DNA, you're going to have a base, a functional group hanging off of the 2' carbon, a hydroxyl always hanging off of the 3' carbon. The 4' carbon has a covalent bond to the oxygen and to the 5' carbon. Hanging off of the 5' carbon are always going to be 3 phosphates unless you lose this and boom, you'll be bound to the adjacent oxygen. So if for funsies here, we're making a new strand of DNA. Let me see if I can make my pen work again. Here we go. I think it's just erasing really slowly. Maybe I'm supposed to use that part. I don't know. You can't see what I'm doing. Sorry. If we say, for example, add another nucleotide here, what we will have is the phosphate, the single phosphate, from the next nucleic acid bound to carbon number 5, which is bound to carbon number 4, which is bound to your oxygen, carbon 1, carbon 2 and 3, and then your base. I'm just going to put a B here. Off of carbon number 2, you're going to have a hydrogen if you're doing DNA. Alright, that looks really bad to be DNA. My apologies. I don't know why this is misbehaving so badly. Let me try this pen. DNA. Alright. Anyways, hopefully this helps a little bit. Definitely watch the videos that I just posted about replication and about histone packaging. One of the biggest mysteries of DNA science was how do you get all this information into an area inside of a cell that you can't even see with the naked eye. And not only are you putting 1 chromosome in there, you're putting 46 in humans, 76 in dogs, right? So this packaging problem and expression problem for functioning of a cell, it has been figured out, and here's the beginning of it. I will hopefully see you guys soon. Bye-bye.